Hi, everyone. Welcome to OsteoBites. Thanks for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Kelly Makielski and Dr. Jaime Modiano from the University of Minnesota College of Veterinary Medicine and the Masonic Cancer Center. And they will discuss comparative extracellular vesicle biomarkers for osteosarcoma risk and prognosis, sharing results from the COED study and implications for the KIDsCAN study. Thank you so much, Dr. Makielski and Dr. Modiano, for joining us in OsteoBites today. Everyone, please feel free to submit questions during the presentation through the Q&A feature on Zoom, and then we will share those with Dr. Makielski and Dr. Modiano after their presentation. A little bit more about our guest today. Dr. Kelly Makielski is an assistant professor of small animal internal medicine at the University of Minnesota College of Veterinary Medicine and Masonic Cancer Center, and her research focuses on extracellular vesicle biology and comparative oncology, using naturally occurring cancers in dogs to inform human cancer biology and treatment. She is a recipient of an NIH KO1 investigating EV-based biomarkers for osteosarcoma prognosis in pediatric osteosarcoma, with the goal of guiding personalized therapy and reducing treatment-related morbidity. And Dr. Jaime Modiano holds the Alvin and Jean Perlman Endowed Chair of Animal Oncology and is the director of the Animal Cancer Care and Research Program of the College of Veterinary Medicine and the Masonic Cancer Center. He completed his training through the Veterinary Medical Scientist Training Program at the University of Pennsylvania, and he followed it with a residency in clinical pathology at Colorado State University and a postdoctoral fellowship at the National Jewish Center for Immunology and Respiratory Medicine. And before joining the University of Minnesota, he served on the faculties of Texas A&M University and the University of Colorado Health Sciences Center. Dr. Modiano has also worked in the private sector as a founder of several startup companies and is director of cancer immunology and immunotherapy for the Donald Monk Cancer Research Foundation. And through his research, Dr. Modiano seeks to understand how and why cancer happens and to develop strategies for improving the health and well-being of companion animals and humans alike. His research has been supported without interruption by federal and private sources for over 30 years, and he has co-authored several hundred scientific papers, abstract presentations, and book chapters focused on immunology, cancer biology, genetics, and diagnostic and therapeutic innovations for cancer and immune-mediated diseases. We are thrilled to have both of you on OsteoBites today. Thanks so much for joining. And before we get started, a few announcements. We actually have several because there's so much going on this October. MIB Agents has funded $2.5 million in osteosarcoma research, supporting 32 investigators through our Outsmarting Osteosarcoma Research Grant Program since it started in 2017. We have two funding mechanisms. There's a $100,000 one-year HERO grant and a $50,000 one-year HOPE YI grant. And the grant period is July 1st, 2026 through June 30th, 2027. The deadline to submit an LOI is tomorrow, October 24th at midnight Eastern time. So there is still time to submit. I'm going to put a link in the chat where you can learn more and find the application link on Proposal Central. We are also now accepting abstract submissions for our Factor 2026 Osteosarcoma Conference, which is going to be June 25th to 27th in Columbus, Ohio, which is not too far from you, Dr. Makielski and Dr. Modiano, so I'm hoping we're going to see you there this year. Abstracts will be considered for both oral and poster presentation, and I'm going to add a link in the chat with more info on that. And I also wanted to share a call for submissions for a special journal issue of Discover Oncology. The special issue is going to be called Making It Better for Osteosarcoma, Translation Research, Comparative Oncology and Beyond. It's edited by Dr. Alicia Valoria-Petit from the Ontario Veterinary College at the University of Guelph. And this collection invites studies that aim to identify those key tools we are currently lacking to make it truly better for osteosarcoma patients. Translational and comparative oncology research involving canine and human osteosarcoma is of particular interest, but they would also like to go beyond to incorporate the voice of patients, their families, and advocates. So therefore, studies at the interface of sociology, psychology, and oncology are also welcomed and encouraged. And altogether, they hope this collection will provide an overview of what all the parties involved are doing to not only improve outcomes, but to enhance the quality of life for osteosarcoma patients. The submission deadline is May 4th, 2026, so lots of time, but just wanted to plant that seed. And I'll put a link in the chat where you can find more info. Lastly, whenever we do a comparative oncology episode, I'd like to point out that MIB has a canine osteosarcoma trials resource on our website. The COED study that we'll be learning about today is also listed there. And you can see a list of veterinary oncology programs in the US and Canada, as well as a list of active clinical trials for dogs with osteosarcoma and contact information if you're interested in learning more about those studies. And I'll put a link in the chat as well to find more information about that. So thanks for bearing with me through all those announcements. Let's go ahead and start with the presentation today.
Great, thank you. Thank you so much for the introduction and for I'm so excited to be able to share my my projects in this to this audience. So before we start, I just want to mention a few disclosures that are relevant, both for myself and for Dr. Modiano, and to mention that these relationships are reviewed and managed by the University of Minnesota annually in accordance with its conflict of interest policies. And also to mention that diagnostic tests for veterinary patients, such as the one that we'll be discussing today with the COED project, do not fall under any regulatory scrutiny in the United States. So it really is up to the user, the owner or the veterinarian to do their due diligence when deciding to do diagnostic tests. So our take-home points after we spend this time together here today are that canine osteosarcoma can be used as a model for certain aspects of the disease in humans, and that liquid biopsies can be used for assessment of cancer risk, early detection of cancer, and prognostication. And this approach from all angles will allow for tailoring prevention and treatment with the goal of minimizing patient morbidity without negatively impacting survival. So the plan for today is to talk about some background, and then I will introduce the KIDsCAN project, which is our pediatric osteosarcoma study, and then the COED study, which is our canine osteosarcoma project. And then I will wrap up with some conclusions, and then hopefully we'll have some time for good questions and discussion. So starting with some background, osteosarcoma is a common cancer in the dog, and there are a lot of features that are similar to the human counterpart. So one of the things that's similar between the two species is shown here is that even though there are some differences in both species, osteosarcoma is diagnosed most commonly in the long bones of the appendicular skeleton. And osteosarcoma is diagnosed very similarly. And in fact, if you look at a canine osteosarcoma tumor under the microscope and a human osteosarcoma tumor, you really can't tell what species it came from just by the microscopic appearance. We treat osteosarcoma fairly similarly in the dog with surgery and chemotherapy. And then in both species, progression occurs in a similar manner with spread to the lungs occurring most commonly. However, in the dog, osteosarcoma is more common, and the progression time, so the time between diagnosis and spread of the cancer, is usually compressed because of differences in lifespan, with median survivals of about a year in dogs. We often diagnose osteosarcoma, unfortunately, at advanced stages. And so this is a radiograph of a dog showing a osteosarcoma tumor in the hind limb. So this is the spine of the dog, and then these are the back legs. And then this is the tumor here, and you can see lysis and proliferation of the bone. And we often diagnose it when it's quite advanced, and so the tumor burden at the site is quite large. And it frequently metastasizes, with the lungs being the most common place of metastatic spread. And so this is a thoracic radiograph here, and this is the spine, and then everything in here is the thoracic cavity, so the lungs and then the heart. And these small nodules that are too numerous to count, that are white and opaque, these all are metastatic spread. So small nodules where the cancer has spread to the lungs, despite this particular dog having received amputation of the affected limb and then follow-up chemotherapy. And so the rationale for our studies is that by assessing which patients are at highest risk of cancer development, by diagnosing osteosarcoma earlier, and by using rational tests to predict response to therapy, this will provide additional options for prevention or treatment, potentially improving patient outcomes, both in humans and in dogs. There are a couple of differences, and one of those that I wanted to highlight is that osteosarcoma occurs more commonly in older dogs. And so this graph here shows the number of cases out of the total on the y-axis, and then the age in years on the two x-axes, with human age in years on the bottom and canine age in years on the top. The dog ages go from 0 to 14, and the humans go from 0 to about 100. And it shows the number of cases out of the total number diagnosed at each age. And so the human data are shown in this solid black line, where you can see that there's a large peak where osteosarcoma is diagnosed most commonly in adolescence. And the dogs, which are shown in the dotted line here, we do see a small peak in adolescence at around 2 years of age, but the majority of our cases in dogs are diagnosed between 6 and 10 years of age. That is one difference that's important to highlight. And then I also just wanted to mention that osteosarcoma is diagnosed most commonly in large and giant breed dogs. And so we see that in the larger the dog's body size, the greater the osteosarcoma risk, and actually the shorter the life expectancy. And the converse is true. So the smaller the dog, the lower the osteosarcoma risk and the longer the life expectancy. And this is a cute figure, but it's also based on actual data. So this Rue paper is a retrospective study that evaluated osteosarcoma risk in various breeds of dogs. And so that's what was used to assemble this figure. And one of the exciting parts of our COED study is that we're enrolling these large and giant breed dogs prospectively. So we are planning to see if we can show what's been shown retrospectively in a prospective cohort of dogs. So what is a liquid biopsy? That's the other part of the background that I wanted to go into briefly. But a liquid biopsy refers to a non-invasive blood test that provides important information about a site distant in the body. And so let's say there's a tumor present in a distant organ. We can actually obtain a blood sample and analyze different material that the tumor cells secrete into the blood and use that for our liquid biopsy test. So it tells us information about the distant site where there's a tumor developing. And there are several advantages of liquid biopsies, including that they are non-invasive or less invasive, and they tend to be less expensive, of course, than surgery. And we can use them to detect tumor material from faraway sites, and that includes sites that are difficult to get to with surgical approaches, or potentially when there are multiple different tumor sites. However, there are some disadvantages, and that can be that the assays can be technically challenging. There can be concerns with reproducibility and with inconsistent shedding by the tumor. So a liquid biopsy test that's positive one day may not be positive if you were to submit it to a different lab or if you were to obtain the sample on a different day. And even though they are less expensive than surgery, they can be relatively expensive for blood tests. And then, as I mentioned at the beginning, in our veterinary patients, the lack of diagnostic regulatory oversight really means that products can be released to the market before they are really ready to be used in a broad sense of the test. And then I just wanted to highlight that there are several potential applications of liquid biopsies throughout the cancer journey. We think about liquid biopsies a lot for early detection, but there are other places where we can utilize them. And so if you think of this little blood vessel as a timeline in the patient's journey, we have a time point where a tumor starts to develop, and then that tumor starts releasing material into the bloodstream. And then over time, that tumor can spread to the lungs. And we can use liquid biopsies to determine risk assessment in addition to early detection. And this refers to a situation where the patient has not yet developed a tumor, but they are at an increased risk of a particular type of cancer. And so the liquid biopsy test can actually be used to predict which patients are more likely to have cancer develop. And then once a cancer actually has developed, we can use liquid biopsies for early detection. So that would be before that the tumor has started to grow, but clinical signs are not yet apparent. And then we can use liquid biopsies for guiding therapy. So looking for particular markers that may predict what patients might be more likely to respond to specific therapies. And then finally, let's say we have our patient that was treated for the tumor and goes into remission. We can use liquid biopsies for disease monitoring, so looking for recurrence or spread of the tumor. I just want to mention that because even though we call our COED study the Canine Osteosarcoma Early Detection study, it's actually probably more in the risk assessment category here. There are multiple different analytes that can be utilized for liquid biopsies, and I've certainly heard a couple of presentations regarding various liquid biopsy analytes in the OsteoBites series. I'll be focusing mostly on EVs because that's where our work lies. And these are extracellular vesicles, but other analytes that can be used include circulating tumor cells and circulating tumor DNA, among others. EVs are these small packages that are released into the bloodstream, and they're actually released by all cells in the body, and they contain important information that can be used to learn about the cell that released them. And the cargo that's contained in EVs can include things like nucleic acids, proteins, lipids, and they're utilized by cells both as kind of like a waste system, so to get rid of things that the cells don't need any longer, but also they can be used for communication with cells downstream. So they can be released into the bloodstream to communicate with a cell in a distant site to potentially change whether or not that location would be hospitable to a metastatic spread. There are some limitations to the use of EVs, the biggest one being that they are released by all cells in the body. And so it can be challenging to differentiate disease-specific EVs or disease-specific cargo from the much larger background of cargo in EVs that's released by normal cells in the body. So essentially, it can be challenging to locate the needle in the haystack. But we've developed this diagnostic platform that can overcome that limitation using a xenograft mouse model. And so essentially what we did was we established canine osteosarcoma xenografts by injecting canine osteosarcoma cell lines into mice. We let the tumors establish, and then we obtained serum samples from the mice at regular intervals. We enriched for EVs from the serum samples and then sequenced the EV cargo. We then aligned the cargo to a combined genome of the mouse and the dog, and anything that aligned only to the dog and not to both species, or not only to the mouse, we knew had to originate from the tumor cells. Otherwise, the mice should not have circulating dog-specific transcripts. So it really allowed us to kind of use the mouse model as a way to filter out relevant disease-specific information. And of course, we wanted to make sure that the biomarkers we identified were relevant in our species of interest. So we then performed in-species validation by obtaining serum and plasma samples from dogs with osteosarcoma, dogs that were healthy, and then dogs with diseases that were not osteosarcoma. And then by applying machine learning, we were able to essentially come up with a gene signature that was able to be evaluated in dogs with osteosarcoma after therapy. And so what we found is that when we had dogs with osteosarcoma that were treated, and we had pre-treatment and post-treatment samples, in the dogs where our machine learning was able to still detect our osteosarcoma signature, those dogs had a worse prognosis after therapy, as shown in this teal line here, where this is a Kaplan-Meier curve showing progression-free survival. And each time a dog is diagnosed with progression of its disease, you see this step down. And so you can see that the dogs shown in here in teal have a worse prognosis up to 15 months after therapy than the dogs shown in this kind of red color, where our machine learning algorithms could not detect our osteosarcoma gene signature after therapy. And we believe that this is likely due to the ability of this gene signature to detect minimal residual disease that's present after treatment. And this work is published, and we are now applying similar methodology to our two ongoing osteosarcoma projects. And I'll talk about each of these, the Canine Osteosarcoma Early Detection Study, or COED, and then KIDsCAN, which is our molecular signatures of biological behavior in pediatric osteosarcoma. And for both studies, we've optimized our methods for processing samples. All of our samples are serum or plasma, and we enrich our EVs from these samples. And then all samples undergo nanoparticle tracking analysis, which is a way to measure the size and concentration of enriched particles. And then all of our samples then will be undergoing RNA sequencing. A subset of our samples will also undergo Western blotting and electron microscopy as further methods of characterizing the EVs that we enrich. And I just want to show what the NTA or nanoparticle tracking analysis looks like. And so essentially we run our sample through a fluid tube, and a laser on the NTA is shown through. And based on the Brownian motion of the particles, as shown here, when the laser bounces off, we're able to determine the size and the concentration of our particles. And I'll be showing some NTA data that was obtained through this method later on. Now moving on to discuss each of the projects. So for each of these, KIDsCAN and COED, I will be going over the goals of the project and a little bit of the methodology, and then some preliminary data. Both of these are very much ongoing projects, and both have sequencing that's in progress. And so primarily I'll be focusing on some NTA data and then also some demographic data in our canine study. But starting with KIDsCAN, this is our pediatric study. Our goal with this is to identify predictive gene signatures in pediatric osteosarcoma. So if we are able to identify a gene signature panel that could predict prognosis, a test like that could be utilized clinically to guide therapy and improve management of this disease. And ultimately, our goal is to identify which patients are predicted to have a better prognosis, where treatment de-escalation could be implemented, with the goal of minimizing the lifelong morbidity that many patients have when treated for pediatric cancers. And of course, we're quite a bit away from that now, but that's our ultimate goal to improve the whole patient experience. And we're doing this through two aims. Our first aim is to sequence serum and plasma EVs from pediatric patients with osteosarcoma that have known clinical outcomes. And so we're working with the Children's Oncology Group, or COG Biobank, to obtain serum and plasma samples from pediatric osteosarcoma patients where we know whether they experienced metastatic spread at a certain time point after treatment. And then we're going to sequence the EV cargo and determine genes and other features that are associated with increased propensity of experiencing metastatic disease. And then for the second aim, we're doing a very similar methodology to what I described in the published dog study, where we're sequencing serum EVs from xenograft mouse models using pediatric osteosarcoma cell lines that have differing metastatic propensities. So we establish our xenograft models, and as tumors establish, we obtain serum samples every couple of weeks. And then we follow the mice to determine which go on to develop metastatic disease. And then we'll go back and sequence the EVs from the serum samples to determine features that are associated with metastatic progression. Here is our panel of cell lines that we are utilizing. All of these are human osteosarcoma cell lines that are pretty well characterized in the literature. And we selected cell lines that predominantly had kind of differing metastatic propensity, so low, intermediate, and high, but also taking into account different distributions so that we had good representation of different characteristics of the patients from which they are derived, including sex and age, ethnicity, and then the morphologic subtype of osteosarcoma. And we did this in an effort to best capture as much of the heterogeneity that we can see with osteosarcoma as possible. So the mouse studies are in progress, and it's been an ongoing journey to obtain our samples from the Children's Oncology Group. Of course, these are very precious samples given the rarity of this cancer, and so we've first obtained 15 samples that have allowed us to compare interchangeability of serum and plasma to know going forward which sample type we want to utilize when both are available, and to make sure that our results that we get when we have differing sample types are as robust as possible. So these first 15 samples we received, this is the age at diagnosis of the patients of these 15 samples. And we had 10 male patients and 5 female patients. Shown here. And then we did some comparisons using nanoparticle tracking analysis to look at serum and plasma. And overall, we found that both size, and then I'll show concentration next, were very similar between the two sample types. So this is nanoparticle tracking analysis, or NTA, showing the EV size. You can see that there's not much of a difference between the two sample types. And then this is a comparison here showing good correlation. And then same thing with the concentration as determined by NTA. So again, very similar results, whether we're using serum or plasma. And these are paired samples from the same patients, I should mention. And then similar comparison between the two. And so this is allows us to work with either sample moving forward. And in addition to these nanoparticle tracking analysis results, we also evaluated for expression of various genes in the EV cargo using QRT-PCR. And then sequencing of these samples is ongoing. Moving on to COED, this is also very much an ongoing project, but we do have a bit more result as far as the different demographic patient characteristics that I'll be discussing, and also some more NTA. So our goal for the COED study is to determine genes associated with an increased risk of osteosarcoma development in dogs that are already at an increased risk of developing this disease. So we obtain our serum and plasma samples from dogs when they are clinically healthy. We enrich for EVs and sequence the contents. And then we are following these dogs out for the remainder of their lives with clinical surveys every 6 months to determine which go on to develop osteosarcoma and which do not. And we have two parts of COED that ran concurrently. COED 1 is expanding upon our machine learning training set. So this is obtaining samples from dogs with known diagnoses. So we had 4 disease categories: osteosarcoma, other cancer, musculoskeletal disease that was non-cancerous, and then healthy control dogs. To be enrolled, dogs had to have a confirmed diagnosis of cancer to be in the top 2 groups, and they had to have evidence of gross disease at the time of sample collection. So either these were pre-surgical or at time of tumor recurrence. And to be in the musculoskeletal group, they had to have a confirmed diagnosis of a non-cancerous musculoskeletal disease. And then to be in the healthy control group, they had to be within the ages of 2 and 4 and otherwise healthy with no history of cancer. And this is just showing one of the cases that was enrolled in our osteosarcoma group on that x-ray. And our goal for this was at least 150 dogs, which we reached. And then we're adding that to about 100 cases from a biobank that we have. And then again, this is our machine learning training set to further train upon what gene signatures are associated with each of the disease categories. The musculoskeletal group was actually added on after the study had originated as a veterinary student project. And we were able to enroll 50 extra dogs in that group that had the non-cancerous musculoskeletal diseases, which is really exciting because it's a very clinically relevant group. Dogs that present with osteosarcoma are going to present to their veterinarian very similarly to dogs with musculoskeletal disease. They're often going to have pain, joint pain or bone pain, and they might be limping. And so it's a very relevant population to have a test available to try and differentiate what's going on. And it also is something that we wanted to make sure that any genes that we identify are as specific to osteosarcoma as possible. So by having a group that has a non-malignant bone disease, we're able to make sure that the biomarkers that we identify are not just due to the increased bone turnover, but actually more specific to the cancer. We're really excited to be able to add that group to make our ultimate results more robust. And then COED 2 is our machine learning test set. So these are our healthy dogs. Each of these are at an increased risk, or at least a perceived increased risk of osteosarcoma development relative to other dog breeds. And these included Irish Wolfhounds, Rottweilers, Golden Retrievers, Irish Setters, Leonbergers, and Great Danes. And then we had a 7th category for other purebred or mixed breed dogs to increase the ability to apply our results to a broader population. And for each of these, dogs had to have a confirmed pedigree to be in one of our 6 target breeds. And then the other mixed breed dogs, they did not have to have a pedigree or anything. For our other mixed breed dogs, they had to be of a minimum body weight and body height to make sure that we are still enrolling dogs that are at some elevated risk of osteosarcoma development. All dogs had to be at least 4.5 years of age. We wanted them to be skeletally mature, but at a point where they're just starting to have their risk of cancer increase. But we wanted them to be in good health at the time of sample collection. So no significant comorbidities and no history of cancer diagnosis. We had a goal of 50 dogs for each of these groups, with the exception of Irish Wolfhounds, where we had a goal of 75, and that brought us to a total of 375 dogs. And this is our machine learning test set, where we will be applying machine learning to assign dogs to one of these risk categories to predict whether they are at an increased risk of osteosarcoma development. I do want to acknowledge that there are a couple of confounding variables, and some of these are kind of by the study design, but age is one of them. So our dogs in COED 2, this is our at an increased risk group that's healthy. But by design, we wanted them to be at least 4.5 years of age. And so there is no overlap with our healthy control population, who by our study design was in between the ages of 2 and 4. And then our other groups, our disease groups, had no restrictions on age. And so we tend to see these diseases in older dogs. So we do kind of have these being a little bit on the older side, but there are some differences here that I think we'll just want to take into consideration when we do our data analyses to make sure that whatever biomarkers we're identifying are not just associated with the difference in age between these populations. And this is not uncommon with different liquid biopsy studies, where oftentimes the healthy control population is at a younger age than the test population. It's just something that we want to keep in mind. And then body weight or body size is also another difference. We did not have any size restrictions on our disease groups here, but these diseases are commonly seen more in larger dogs. Our healthy population had a nice wide range because we enrolled all healthy dogs. And then our COED dogs, these were all large and giant breeds. And so unsurprisingly, they did have an increased body weight overall compared to the other disease groups. So that's just another thing worth noting is the difference between our study populations. For COED 2, we enrolled our 6 target breeds in our 7th group from all over the United States. And so this map really just shows our distribution by breed from dogs that were enrolled in COED 2, where the different breeds are shown with different colored dots. And then the number of cases in that geographic location is depicted by the size of the dot. And we wanted to look at this because the study is out of the University of Minnesota, but we wanted to have a good distribution of dogs. We didn't want only Minnesota dogs to be in our test population. And as you can see here, we had a pretty good geographic distribution of our enrolled cases. Age at enrollment did not differ significantly between breeds, and this will come into play because we looked at differences in mortality between the different dog breeds. And so we wanted to see if there were any changes in the age at enrollment. And overall, no major differences by breed as shown here, with again, all of the dogs having to be at least 4.5 years of age at the time of enrollment. There were some interesting differences, not unexpected, but still interesting in body weight between the different dog breeds. So all of these dogs are large and giant breeds, but they were differing, degrees of large and giant. So our Irish Wolfhounds, shown in this coral color, and the Great Danes, shown in purple, are definitely our largest breed of dogs. Those are our most giant, whereas the Rottweilers and Leonbergers are kind of in the middle, and then the Golden Retrievers, Irish Setters, and our other category were on the lower end for body weight. And as I mentioned, we follow these dogs out, so they're healthy at the time we obtain our sample. And then every 6 months from that point on, for the remainder of their dog's lives, we send the owners a survey every 6 months to just ask, is the dog still alive? Have they been diagnosed with a significant disease? Have they been diagnosed with cancer? Have they been diagnosed with osteosarcoma? And when the owner reports a disease diagnosis, then we obtain the medical records from the veterinarian with their permission and use the medical records to validate the reported response. So determine the likelihood that that is a true diagnosis, and then also to assign a level of evidence for that diagnosis. And our survey responses have really exceeded our expectations, with greater than 80% at every time point so far. So these are our survey response rates, and the different time points are shown at the bottom here by 6-month intervals. And then the percentage of cases eligible to receive that survey is shown in the different bars, where blue is the ones that the owners have completed and returned that survey to us, and then gray is the smaller percentage where we have not received a response. And this is out of the number of cases so it's normalized to 100% to reflect the number of cases that were eligible to receive that survey at that time point. So in some of our cases, they have not reached 36 months after enrollment, so they wouldn't have received that survey yet. And in cases that have already died or were euthanized prior to that time point, we don't continue to send surveys. But as you can see, we have greater than 80% at all of our different time points, which we were really pleased about. And another thing we were pleased about is that the majority of our dogs are actually still healthy, despite it being a couple of years now since being enrolled, and despite them being adult dogs at the time of enrollment. So about 2/3 of our dogs remain healthy on our surveys. Of the dogs that have had a reportable outcome, cancer has been the most commonly reported. And this is all causes of cancer, not just osteosarcoma shown here, but that's been our most common diagnosis that we've had reported after healthy. And we were curious about the different ages that the cases that were diagnosed with cancer, what age that they were diagnosed at. As you can see, our peak so far is between the ages of 7 and 9, keeping in mind that not all of our cases have reached 10 and above yet. So there's a smaller number of cases that are even at that age yet. And then this is showing the proportion of dogs that were enrolled in our study by breed that are still alive as of right now versus the ones that have died. So the dogs that are still alive are shown in this green color on the top, and the ones that are deceased are shown in the yellow color on the bottom. And you can see that there are some breed differences, and the numbers are over here on the right if you prefer to look at the actual numbers, where just over half of our Irish Wolfhounds have actually either died or have been euthanized since enrollment, versus the lowest is in our Irish Setters, where it's only 14%. So really just highlights some ongoing need for research into why we're seeing differences in death in these different dog breeds. And again, this is all prospectively collected data. And then in addition to the proportion that have died, we also looked at the age at time of death for the dogs that have died so far. And you can see that in addition to being the ones that have the highest percentage of death, Irish Wolfhounds also had the lowest age at death. And then our other much larger dog breed, Great Danes, is one of the younger ages at death, but still different than the Irish Wolfhound. So we're really interested in kind of what is driving that. And our other breed group, which is definitely more heterogeneous, does have an older age at death so far. One of the things we wanted to investigate is whether higher body weight was associated with a younger age at death. As I mentioned in that figure in the background, that is something that's been reported in a retrospective manner, that the larger the breed of dog, the younger the lifespan generally. And so we were interested to see whether this held true in our prospectively enrolled cohort. So this is showing the body weight in kilograms on the y-axis and the age at death in years on the x-axis, and the different breeds are shown in these different colored dots. And you can see that there is an association where higher body weight was associated with a younger age at death. And the same holds true for increased body height as well, where taller dogs have younger ages at death reported. It's worth noting that these are all causes mortality, so not just cancer related, but all diseases, and also when both natural death and euthanasia were included. But when we separate out cancer versus not cancer deaths, these relationships still hold up. And then moving on to some nanoparticle tracking analysis in the COED study. Again, we can use this to tell us about the physical parameters of the EVs that we are enriching from our samples. And specifically, we wanted to investigate whether we could use these physical parameters, size and concentration, to differentiate between our clinical groups of dogs. So comparing healthy dogs, their EV size and concentration, to dogs with osteosarcoma, dogs with other cancers, and dogs with benign musculoskeletal disease. And what we found was that overall, the groups overlapped quite a bit when looking at size and at concentration. This is the EV size shown here in healthy osteosarcoma, other cancer, and non-malignant musculoskeletal diseases. And due to the overlap in these graphs, you can see that you can't really take one sample and use it to reliably tell you whether a dog is in a healthy group or does it have osteosarcoma. And the same holds true with concentration of EVs, where the groups overlapped quite a bit. And this really tells us that we can't use these physical properties of EVs alone to differentiate between clinical groups. And so sequencing of our EV cargo to determine gene expression is really going to be the way that we're able to differentiate between clinical groups. And this is important to us because we wanted to make sure that whatever results we put out there are as robust as possible so that we can trust our findings. So sequencing is in progress, so we can determine our final biomarker panel. And so these are my lab group members. So we have Adam on the left, Jaren in the middle, and then Julia on the right. And they are hard at work enriching EVs, getting RNA, and then submitting that for sequencing and doing all the other EV things that we do. So with that, I'd like to wrap up with some conclusions and then leave some time for questions and discussion. So our ongoing work, we have optimized our methods, and our samples are currently undergoing sequencing. We're analyzing both clinical data and also the data that we get from our EVs, both the physical data and the sequencing results. And then we will continue to obtain clinical follow-up from our COED 2 dogs every 6 months. And that's very important because, as I mentioned, about 2/3 of our dogs are still reported to be healthy. And really knowing what their end diagnosis is going to be the best way that we can look at the different features in our samples that are predictive of who's at a higher likelihood of developing osteosarcoma and potentially other cancers. So this ongoing clinical follow-up is critical to make sure that we're correctly predicting. And we're currently assembling a manuscript to report our prospective health status analyses for all of our enrolled breeds. So those associations between body weight and age at death prospectively collected. So in conclusion, EVs are these small packages that are released by cells, and they can be enriched from blood and used diagnostically in canine and human patients. And our ongoing work to sequence EV contents should allow us to obtain disease-relevant gene signatures to help inform risk assessment of osteosarcoma in dogs with the COED study, and to inform prognostication of osteosarcoma in our pediatric patients with the KIDsCAN study. And I'll go back to our take-home points. So hopefully we all agree now that canine osteosarcoma has aspects that can allow it to be a model for certain parts of the disease in the human counterparts. Liquid biopsies can be used not just for early detection, but also for assessment of cancer risk and for prognostication. And by having a multimodal approach, this will allow us to really tailor prevention and treatment to the individual patient. And as I mentioned, our ultimate goal is to use these diagnostics to minimize morbidity, but without a negative impact on survival. So really allowing us to target the treatment who really needs aggressive therapy versus who could potentially undergo treatment de-escalation and be spared some of the debilitating side effects that these treatments can have. I'd like to acknowledge these funding sources, and then also acknowledge the members of my lab and the relevant members shown here of the Modiano lab. Thank you to the Minnesota Nano Center, where we bring our samples to do our NTA. The Clinical Investigation Center at the vet school, they help get our samples and enroll our cases in COED 1, and then these collaborators here. And with that, I'd like to thank you again for allowing us to share these ongoing projects in this forum, and I'd be happy to try to answer any questions.
Thank you so much, Dr. Makielski, for walking us
Thank you.
through both of those studies. And we do have some questions that have come through. So let's just start with those to make sure we get to them. Actually, let's start first with questions about the COED study. So just one question about for the surveys, which was an amazing response rate that you got for those surveys. Were those conducted electronically, on paper, another method?
Yeah, great question. We send them out electronically, and so every 6 months, an email is auto-generated with a link to the survey, and then the data are imported into REDCap, and that allows us to just kind of export the data very easily, and it's all standardized. And part of our great response rate is that we do try to keep our surveys very short and sweet. There's a lot of data that would be really interesting to collect, diet, exposures, various things that we're sacrificing to get that good response rate. So we really just want to focus on what's the ultimate outcome of the dog, and then use that to then get records where we can fill in the gaps.
I'm just curious, are you pretty successful in procuring those records? Because I know sometimes in human studies that there's a little bit of a drop off there, and I'm just curious in the canine world how successful you are with records.
I have to give a thank you to Mitzi, who is our sample coordinator and who does all of that record wrangling. She calls our vets and has them fax over the records. And I would say once in a while things fall through the cracks, but for the most part, we do a really good job of getting the records. One of the limitations, though, is that these are all client-owned dogs. They're part of the family. And so owners can decide how much they want to do diagnostically when disease is suspected. And so we don't necessarily have full diagnostic data. A diagnosis of cancer might just be based on a radiograph that looks like a tumor, but maybe they didn't elect to go to biopsy or tumor removal. And so that is a limitation. And we use the records to help us to get all that information and assign a level of evidence.
Gotcha. And then another question that came in about the COED study. Do any of the national and international companion dog cancer omics registries and biobanks offer the ability to increase the richness of your OS data sets, helping to have data science better find which pathways to focus on and target?
That's a great question. I will try to answer, but I'm curious if Jaime has anything to add. I don't think so, unfortunately, because we are utilizing EVs, and that's not something that's been made readily available because it's not the most common source of material. Do you have anything to add, Jaime?
Yeah, I agree. And when we looked at comparing the EV cargo to the distribution of genes in tumor cells, the overlap was really minimal. So whatever mutations or transcriptional programs are present in the cells are not necessarily reflective of what's being packaged into EVs. So we really have to deal with trying to understand what commonalities exist in the EV landscape, which will include not only what's being secreted from the tumor, but also what the host is responding. So it's great to have those registries, but like Kelly said, because EVs are not something that's been routinely sequenced, they're not necessarily helpful. Apples and oranges.
Gotcha. And then a question that came in about the human study, the KIDsCAN study. Why do you need to rely on banked samples instead of obtaining new ones from patients?
Also a
My
great
answer is
question.
like, oh my God.
We would be thrilled to be able to obtain prospectively collected samples for that study as well, but just due to the rarity and how precious these samples are and how many other studies are wanting to utilize pretreatment samples, how much blood can be safely obtained at each time point in the clinical trials, it's very challenging. But it would be really cool to be able to collect those prospectively. It's just not probably feasible.
Yes, samples are definitely difficult to procure.
Yeah, well, not only that, I mean, the IRB process for justification, when you layer on that on top of what everything that Kelly said, the IRB is going to be really reluctant to add more morbidity to the kids when they're already being, collected and sampled and treated to death. So it is a more reasonable approach for the patients to coordinate this through a central group like COG than to try to have everybody doing their own thing, particularly for a disease as rare as osteosarcoma. So it does create interesting challenges, though, in terms of trying to log samples or unlock samples out of this precious bank, which lie between multiple layers of justification, security, very useful comments, and sometimes very whimsical comments from the reviewers that man those logs. So, yeah, but getting through to being able to get samples was a process, and it's also a major one.
Yeah. So, I think we've heard about different studies for ctDNA or CTC liquid biopsies. And wonder if you could just kind of expand a little bit more on how these EV-based liquid biopsies differ from those.
Yeah, that's a great question. And ultimately, each methodology has its pros and cons. And until we have a perfect test, I think all of them are worth investigating. We have selected EVs for a couple of reasons. It's a little bit less explored area in this disease, and especially in the veterinary side of things. So I think there are a lot of unanswered questions still. We also have the experience with the published canine study showing that we were able to get relevant disease information out of EVs. And so it really gave us some kind of foundation for moving forward with that as our approach. But really, I think there's a lot of fascinating work that's going on, and hopefully somebody comes up with a test that can help with this disease, and whatever it ends up being, it has my support.
Yeah, our original design for EVs, which actually preceded Kelly before she took over the project and made some fantastic advancement, was to look at EVs because there's reasonably good data indicating that EVs are part of conditioning the metastatic site. So trying to find signatures that are associated with metastasis might also give us insight into the mechanisms of conditioning the metastatic site. It may or may not, because there's a lot of other factors that play a role, but that was one connection. The other thing to note is that we're triple dipping because we have 3 different studies looking at 3 different methodologies for risk assessment in different diseases. So we had one looking at patterns of cells in the circulation that would inform risk or presence of cancer, which was focused on hemangiosarcoma, but we had multiple cancer types included in the study. We have COED, and then we have a newer study called LYRA, where we're essentially the focus is lymphoma, but we also have multiple different cancers that are coming into the study, where we're looking at cell-free DNA, RNA sequencing, and multiple patterns of chromatin accessibility in the blood. And so because every study contains multiple different tumor types, we are able to understand if the target cancer for that particular study is different, whether that particular method is better for the target cancer or any of the other cancers included. And at the end of the day, we might be able to actually coalesce data and come up with features from all these different things to create a little kit where you can do maybe 3 things, and those 3 things could involve different parts of the studies. So it's nice when a sample collected for one study can actually be applied to 2 or 3 studies. And we ask the owners to allow us to do that, and also to share the samples broadly with other people. So if there are people who are doing any type of investigation that could benefit, to the extent that we have any material left over, we are absolutely happy to share it.
Great. So Dr. Makielski, you talked a little bit about the pros and cons of a liquid biopsy versus tissue, which is even more difficult to procure. And I think, we've learned that with tissue, there is that intratumoral heterogeneity. So, even if you were to get a tissue sample, that might not be representative of the entire tumor or even different metastatic sites. And so are you seeing that in, like, can liquid biopsy, I guess, help solve that? Or you did mention, I think you said something about like inconsistent shedding, or is that also an issue in liquid biopsy, or do you think liquid biopsy can better capture that heterogeneity?
I think I'm also curious Jaime's answer to this too, but I think that it has the potential to capture the heterogeneity, but there are always limitations to any diagnostic test where you're going to be limited by that sample that you got. And so um, different EVs throughout the whole circulation may have different cargo. And if you just don't happen to catch the ones that you're looking for, then that test may be non-diagnostic. And I think it's pretty well established that in both dogs and in humans, osteosarcoma is a very heterogeneous tumor where each patient's tumor is different from the other patient's tumor. There's not really any markers that we can reliably use um, to within this cancer. And so I think those limitations are probably also going to be affecting liquid biopsies. But the machine learning approach can help us to kind of reduce some of that heterogeneity and look for kind of things that are regulated together. And so it will reduce our reliance on a single biomarker by allowing us to look at kind of like a panel of things and hopefully catch more cases that way, would be my response to that, but it's a good question. Do you have anything to add, Jaime?
I'm sorry, I was answering Daniel's question online real quick. So can you tell me what the question was?
About the heterogeneity that is present in osteosarcoma tumors, and are liquid biopsies able to mitigate some of that heterogeneity, or are we going to have that as a limitation for us too? Does that summarize it?
That's a great question, and I think it's one of the things that we have been sort of pounding the drums that because the tumors are so different, trying to find tumor specific markers is going to be really really challenging, and in fact there's no real evidence that the mechanisms that they use to metastasize are uniform. So there's some really nice newer data from Ryan's lab at Ohio State that documents some interactions between the tumor cells and the lung environment that might be generalizable among tumors and also among species. But there's really nothing to say that the tumors don't use different roads to get there. And so, you block one path, they use another. So I think focusing on things that might be coming from not necessarily the tumor, but the generation of this new tissue, the disruption of the tissue, the host response, which tend to be much more conserved, is going to be more valuable. So in the original study, the genes that we found that basically said osteosarcoma present, interestingly, had nothing to do with bone formation, tumor growth. They were essentially genes that are upregulated in cells that get bloated. And so these tumor cells are making a lot of stuff and they get bloated and they need to ramp up their waste disposal mechanism. And those are the genes that were being packaged into exosomes and shipped out into the blood. So the osteosarcoma signature present meant bloated cells somewhere, interestingly. And so I think that it ultimately may come down to these non-tumor specific, but rather tumor response associated mechanisms that will give us the better indicators of what the tumor is doing and maybe some of the vulnerabilities that we can attack, so that we don't have to come up with, one therapy for one patient at a time.
Mm-hmm. Interesting. Okay, so just to clarify, so the KIDsCAN study, so the goal there, it sounds like, is really trying to find some gene signatures that can determine risk, prognosis risk. You had mentioned the main goal is then if someone's at a lower risk, then we don't have to have so much chemotherapy to reduce toxicities. And then, so for dogs, for the canines, it sounded like it was more risk assessment, early detection. And so I'm curious how you see that kind of changing clinical care.
Yeah, that's a great question. I think it also kind of is affected by the prevalence of the disease in the two different species. So, because osteosarcoma is so rare in children, it'd be very challenging and probably impossible to do an early detection test unless you were really targeting to specific like syndromes. So it being more common in the dog, especially dogs with certain features like large and giant breed dogs, allows us to be able to look at genes associated with risk assessment or early detection. And how we would use that is if we are able to really predict which dogs are at an increased risk, those owners can be guided to watch for clinical signs like more vigilantly, potentially do diagnostics more earlier, more screening tests kind of regularly if they choose to. But the real thing is that if we can find some sort of preventative treatment, and I know that Jaime can kind of speak to that a little bit more with the EBAD experience, but the goal would be to be able to use some sort of cancer prevention strategy eventually so that any test that we do is not just another source of anxiety where a positive liquid biopsy test that says your dog's going to develop cancer at an earlier age. Right now, that would just give most owners anxiety about, when that's going to happen in their beloved dog. And so having a way that we can really strategically go in and prevent the tumor from even being formed would be the ultimate goal.
Mm-hmm, mm-hmm.
Yeah, and as Kelly said, we completed a study that's now in follow-up stage for interception using a drug called EBAT. And so that was a feasibility study. We enrolled 15 dogs at high risk in a different study and then followed them to see if, first of all, that we could do this, right? And what the logistics would be, what the cost would be, and then whether we could actually follow the dogs and understand if there was a difference. So I think we got an A+ for the feasibility. It was very doable. Possibly within access of cost for many families. As far as whether the treatment works, so far so good. We are beating the odds, so we're better than a coin flip. But we have to wait until the dogs reach that magical point of 1 year cancer-free, 2 years cancer-free after their initial assessment of high risk and compared to dogs that were not treated to see if the signal is positive. So far so good. I think that the last week, if I remember correctly, was a 3-day, 2-day symposium for St. Jude. Was that I can't remember. It was 2 days or 3 days, Kelly?
I was not, it was 2 days, but I was not able to attend because I was on clinics. Sorry.
So St. Jude hosted a fantastic symposium on cancer predisposition. And a lot of the conversation about trying to understand how to get ahead of cancer in syndromic patients ask these same questions, right? Like how can you do it reliably? How can you, once you have the information, what can you do about it? And so we've been having conversations with Logan to reach out to folks that are dealing with patients who are in families at high risk or who have biorepositories from syndromic families to see if something like we're doing for COED could actually be helpful in early detection and management of that small proportion of kids that would be at risk for developing osteosarcoma because they already carry some predisposing gene like a mutation in P53 or something else. So we've been definitely thinking about that. Like Kelly said, the relatively low frequency of the test and all the negative impact of a false positive means that trying to do this in the general population is probably neither warranted nor feasible. But doing it in a population at risk will probably be something that we'd be interested in doing.
Oh. All right. So I was just taking a peek at the site. So COED Part 1, it looks like is still open and enrolling. And COED Part 2, it says it's ongoing, but enrollment closed. Is that the latest?
I would say COED 1 is also not enrolling new cases.
We won't turn them down if they come, but yeah, we're not. We have made enrollment for the full study. So now, the samples are being sequenced, and so anything that we get now will essentially be sort of stored and added for later validation.
Okay.
So,
But you did have participation across the country, but the study is do people just mail in their samples?
we so Mitzi, who was the goddess of recruitment, would put together a kit, coordinate the visit with the owner and the veterinarian. She would send a kit in advance for the veterinarian to have everything on hand to draw the blood. Kelly put together some beautiful instructions on blood draw so that we would get clean sticks and good samples. So everything was right at the hands of the vet or the vet tech. They would then drop the samples back in the box in a prepaid FedEx box that would be shipped to us. And this one was challenging because we had one box that had to come room temp and one box that had to come cold. So there was
That's Shine On.
Oh, that was Shine On.
Everything was cold for
Everything was
COED.
cold. Okay, so yeah, it's I get confused with all these different things we're doing, but Mitzi keeps them straight, and she's amazing. So if you've ever interacted with Mitzi, you have no idea how many emails I get from people saying that she was the sweetest, most empathic, most wonderful person in the world, helping them manage their anxiety and their stress and participating in these studies. So it's
Wow,
really,
I feel like I need to meet Mitzi now.
You do, and sadly, she's telling me that she's going to retire. So,
oh no! Okay, I'm gonna reach out like ASAP. Well, what a great model though too for a local like a one institution study, but able to get national enrollment. That's really impressive, and that's actually, I think it would be great for others to kind of learn from that model. Mitzi has to share her secrets before she retires. Well, thank you both so much for joining us today, Dr. Makielski and Dr. Modiano, for joining us today and for making it better for both our 4-legged and 2-legged osteosarcoma patients. More information on this and all OsteoBites can be found on our website at mibagents.org, on our MIB Agents YouTube channel, and at your favorite podcast place. And then we hope you can join us next week. We're going to be joined by Dr. Jing Huang from the NCI to discuss his work studying the epigenetic regulation of the type 1 interferon pathway in osteosarcoma and implications for immune checkpoint blockade. And actually, we hope that Dr. Huang can actually join us because of the government shutdown. So that hasn't been resolved yet. So we'll see. We'll send an update next week. But if you're interested in that episode, if you just register, even if it does get postponed, you'll then get the first heads up about when that new date is. So if you're interested, still please register. And then you can find our OsteoBites lineup for the next few months on our website. Please subscribe to our YouTube channel so that you never miss an episode. And if you ever have any ideas for future topics that you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you so much again, Dr. Makielski and Dr. Modiano, for joining us today. And thanks to all of you for taking time out of your day to join us. We hope to see you and Dr. Huang back here next week on OsteoBites on October 30th. Thanks so much.
Are we allowed to say, please contact your senator and representative and tell them that we want the shutdown to end?
Sure. I think that is not partisan at all. I think we all want.
It's not partisan at all. Yeah. Let's get our government working again. So,
yes.
Thank you.
Thank you. Thank you both.
Bye.
All right. Bye-bye.